The next study is also looking at maternal activation of the PWS region genes using a different approach. This is a year 2 study. So this group at Duke University has previously used a CRISPR-based approach to do maternal gene activation in PWS. And they showed that they were able to reactivate the maternal genes in about 20% of PWS cells. And that's one question we have not yet answered as a field, is like, what percent of activation needs to happen in order for there to be benefit? But this group is really focused on, okay, so how do we get that percent up? And then how do we maintain expression in the long term? Because those are questions that are going to need to be addressed if we're going to do genetic therapy. So in year 2, they're going to determine the epigenetic requirements for a uniform and stable reactivation of these genes in human cells using these CRISPR-based approaches. We're excited about this therapy because, like any epigenetic therapy, it has the potential, at least, to address all of the symptoms of PWS in one fell swoop. We've yet to know that for sure, but at least the potential is there. This is a very young, enthusiastic researcher who has done a lot of work and was very productive in year 1 of the project. And she is in a larger lab that is one of the leaders in using CRISPR technology to activate genes. So they have done a tremendous amount of work sort of advancing the general field of how you use these molecules to the best effect. So this project will lay the groundwork towards therapeutic development of a one-time transient epigenetic therapy with the goal of stably reactivating the PWS genes for the lifetime of the patient.